Hello students, in last lecture we discussed about the fluorescence microscopy, and with that we have finished all the types of light microscopy. Today we are going to discuss MCQs on light microscopy. First MCQ, the first microscope was invented by option A Leeuwenhoek, option B Hooke, option C Janssen, option D Sheldon. The answer to this MCQ is Janssen. Zacharias Janssen was the first man who invented the compound microscope. Leeuwenhoek invented the simple microscope, and Hooke was the first scientist who used the compound microscope. But original inventor of compound microscope is Zacharias Janssen. Next MCQ, father of microscopy and microbiology is A option Theophrastus, B Janssen, C Leeuwenhoek, D Hooker. The answer to this MCQ is Leeuwenhoek. So Leeuwenhoek is the first scientist who observed the live microorganisms under his own simple microscope. He is the inventor of simple microscope, and that's why he is called as a father of microscopy and father of microbiology. Next MCQ, microscopes having two eyepieces are called A option uniocular microscope, B binocular microscope, C multiocular microscope, and D confocal microscopes. So answer to this MCQ is binocular microscopes. We know that eyepiece is also called as ocular. Uniocular means microscope having single eyepiece. Binocular means microscope having two eyepiece, and multi means having many, and confocal microscopy is one of the type of advanced phase contrast microscopy. तो जो माइक्रोस्कोप में दो आईपीस यूज होते हैं उस माइक्रोस्कोप को कहते हैं बाइनोकुलर माइक्रोस्कोप्स. Which microscope is used to observe living unstained cells? A option bright field microscope, B fluorescent microscope, C dark field microscope, D electron microscope. So answer to this MCQ is dark field microscope. We know that in dark field microscope dark field stop is used, and due to the presence of dark field stop or opaque disc, only the light refracted by the specimen is transferred towards the objective lens, and here there is no, and in dark field microscopy there is no necessity of staining procedure, and therefore we can observe the living unstained cells. Next MCQ, the maximum resolving power of a light microscope is A option 0.2 nanometer, B 0.2 millimeter, C 0.1 millimeter, and D 0.2 micrometer. So option to this question is D. Answer is D. So we know that resolving power is nothing but the ability of the microscope to show the two near object distinctly separate, and the resolving power of the light microscope is limited. It is only 0.2 micrometer. Chemical compounds that are used to stain in fluorescent microscopes are A option crystal violet, B fluorochrome, C methylene blue, D safranin. So the answer to this question is fluorochromes. We know that fluorochromes are nothing but the fluorescent dyes which are used to stain the specimen in fluorescent microscopy. The simple microscope has the resolution of A option 0.1 millimeter, B 1 millimeter, C 10 millimeter, and D 100 millimeter. The answer to this question is 0.1 millimeter. Next question, a compound microscope has how many lenses? Option A 1, option B 2, option C 3, option D 4. The answer to this question is 3. Compound microscope have 3 lens system. First is the condenser lens, second objective lens, and last one is the eyepiece lens. Eyepiece also called as ocular lens. So these 3 lens systems are present in the compound microscope. Next question, which of the following light is suitable for getting resolution? A option red, B green, C blue, D orange. The resolution of compound microscope depends on the wavelength of light used and the numerical aperture of the lens system. Shorter the wavelength, more will be रिजोल्यूशन। तो यहाँ पे ब्लू लाइट जो है, ब्लू वेवलेंथ की लाइट, उसकी वेवलेंथ शॉर्टर होती है। और ये जो तीनों है, रेड, ग्रीन और ऑरेंज, इनकी वेवलेंथ क्या है, ब्लू लाइट से ज़्यादा है, इसलिए आंसर क्या है, ब्लू। ऑप्शन सी इज द आंसर। नेक्स्ट क्वेश्चन, व्हेन the power of ocular lens is 10x and objective lens is 20x, the magnification is. The magnifying power of microscope is determined by multiplying the magnifying power of ocular lens into magnifying power of objective lens. तो यहाँ पे ocular lens का magnification कितना दिया है, 10x. 10x means 10 times, 10 multiplied by 20. 20x means 20 times. So 10 multiplied by 20, answer will be 200x, means 200 times magnification will be obtained when 10x ocular lens is used and 20x objective lens is used in the microscope. So the answer to this question is 200 times, option C. All the following are components of compound microscope except A option stage clips, B fine adjustment, C electron gun, D binocular eyepiece. Answer to this MCQ is electron gun, because electron gun is present in electron microscopy, and all these three, stage clips, fine adjustment, binocular eyepiece, are the components of compound microscope. Next question, the resolving power of unaided human eye is 100 micrometer. Answer to this MCQ is 100 micrometer. Next question, all the following are the components of compound microscope except A option stage clips, B fine adjustment, C electron gun, D binocular eyepiece. So answer to this question is electron gun, because electron gun is present in electron microscope, and all these three are the components of the compound microscope. Now next question, the resolving power of unaided human eye is. Answer to this question is 200 micrometer. Which of the following is used to visualize live cells? A option SAME, B option TAME, C option phase contrast microscopy, D all of these. What is the full form of SAME? SAME means scanning electron microscopy, B transmission electron microscopy. So here the answer to this MCQ is phase contrast microscopy. In phase contrast microscopy, live cells are observed and we can determine the accurate size of the different types of living cells. Next question, the refractive index of air is. A option 1, B 4, C 0.5, D 2. So here the answer to this question is 1. So refractive index of different medium is used to determine the numerical aperture of the lens system. So here the refractive index of air is 1, and that of the oil immersion objective have refractive index 1.56. Remember this, because immersion oil is used when we use the oil immersion objective in compound microscope. Thank you.